In this video, we're going to talk about the structure of the hypha of the fungus and also a little bit on general ideas on their reproduction. So remember that the body of the fungus, called the mycelium, is made of individual thread-like structures called hyphae. These hyphae have cell walls that are strengthened with a carbohydrate called chitin. So that's one distinguishing feature of fungi. For example, plants have cell wall made of cellulose. Fungi have cell wall made of chitin. Bacteria have cell wall made of peptidoglycan, right? So the component of the cell wall is also an identifying characteristic of the kingdom. Most fungi have their hyphae divided into cells by divisions called septa. So if you look at this hypha here on the left-hand side, this hypha has this little kind of coin-shaped structures that you're seeing there, and each of these structures is called a septum or plural septa. Not all hyphae will have it, but most of them will. And these septa separate the cells. However, they're porous. So they're very unique and very different from other organisms because they're porous enough that you can have nucleus going through, mitochondria, so you can have ribosomes. You have a lot of the organelles can just go from one cell to another. So fungus is very fluid. We call this hypha with the septa septate hyphae. Although there's one group of fungi that don't have septa. Instead, their hyphae are like this. They are multinucleated, meaning that there are no separations between the cells. When there is cell division, there is no cytokinesis. That's pretty much it. Okay, so it goes through the whole process, telophase. When it gets telophase, it just now goes into interphase, and then it's going to divide again. So it's going to create this multinucleated structure, the hypha. We call these hyphae cenocytic hyphae. This is the name right here in the bottom. Okay, so cenocytic hyphae as opposed to septate hyphae. Now, the hyphae that have septa can kind of be in two different stages that we call monokaryotic, and karyo means nucleus. So monokaryotic stage would be a stage that there's only one nucleus, and the dikaryotic stage, which would be a stage with two nuclei. However, as I mentioned before, the septa between the cells is porous, right? So there's the nucleus going back and forth. So you will see at any point in time that one of these partitions on the hypha may have one nucleus, may have two, may have none, may have more than two. However, what this means, monokaryotic versus dikaryotic, is actually related to one reproductive stage of the fungus. When they reproduce sexually, so if you look at this diagram here, imagine this is a spore of a fungus, this round thing at the tip, and then when it germinates, it produces the hypha. Now notice that the hypha that's being produced here is septate, right? And it has these nuclei that are in this one on the top. They're shown in black. If you look at the fungus in the bottom, it's shown in white. As these hyphae grow, if these two different fungi from different mating strains come in close contact, they're going to fuse those hyphae during the beginning of sexual reproduction in fungi. However, in fungi, it's very different from other organisms again. That's why they're fascinating. They're just so unique. The nuclei of the two parents don't fuse right away. Okay, so we actually differentiate fertilization into two processes. One of them called plasmogamy, and then the other one called karyogamy, which is not shown in this diagram. Plasmogamy will be the first step of fertilization, okay, but there's no fertilization yet. What happens in plasmogamy is that, think of cytoplasm, okay, is that the cytoplasms, okay, so these two hyphae from different fungi will fuse into one. However, the nuclei don't fuse, so there is no fertilization yet. During this stage here of sexual reproduction, after they fuse, now the hypha that's common to both, that's going to be formed, is a dikaryotic hypha, because you can see here that it has both nuclei from each of the parents, right? The black representing the one on the top, the white nucleus representing the nucleus from the bottom. And we'll stay like this for a while. So in mushroom producing fungi, this will form the mushroom. So the whole mushroom is made of dikaryotic hyphae. So they have a very fascinating type of sexual reproduction. First, the haploid phase is the dominant phase. The only phase of the life cycle of fungi that is diploid is the zygote. So if you look here in this diagram on the right, if you don't remember alternation of generations, I recommend you go back to the plant module and study that again, because fungi have a type of alternation of generation in which you have an alternation between diploid and haploid. However, there's a third stage, as you can see here, right? So you have here on the top representing the diploid stage, which is 2N, and then you have the N stage, which is haploid, but you also have a stage that is the N plus N stage, which is that dikaryotic stage. Fungi have an interesting thing, which is they produce spores, and they can produce spores in two different ways. They can produce spores via mitosis, which the resulting spores will be called asexual spores, or they could produce spores via meiosis, which the resulting spores will be called sexual spores. So as I mentioned before, the mating itself has kind of these two steps, so fertilization is not immediate. First one is plasmogamy. So let's take a look at this diagram here. This is a mushroom producing fungus. Not all fungi will be the same. This is one phylum of fungi. The spores are produced by meiosis, remember, in sexual reproduction. So sexual spores are going to be produced by meiosis in these structures here, which are called in general sporangia, right? So structures that produce spores. These spores are here at the tip. They're going to get out, be released, and they're going to land somewhere. And then they're going to germinate and form a hypha. So this part here, from spore to the hypha, is haploid. Then this hypha, and it's going to form a mycelium, it's not showing here. It could form a mycelium and live for a long, long time before mating. But when it comes to mate, these two hyphae are going to fuse. Remember that they fuse, but the nuclei do not fuse. So they fuse the cells and the hypha, but the nuclei do not fuse. This process is called plasmogamy. Plasmogamy, the fusion of the cytoplasm of two mating types. It will form a dikaryotic mycelium. If you look at the mycelium here, the dikaryotic mycelium, you'll see that between the two septa, you're going to have two nuclei in there. This dikaryotic mycelium is going to grow into a fruiting body. So it's all dikaryotic, right? So we call this phase N plus N. And if you look at the mushroom at the bottom of the cap are these lines called the gills. At the tip of these gills, there are these club-shaped structures, which are the sporangia. And inside, so if you think of this being the cell of the sporangium, it's called a basidium in this group, it's dikaryotic. But in there, in this one cell, these two nuclei are going to fuse. And when they fuse, that's fertilization. We call this process karyogamy. Karyo, nucleus. So gamete is fusing. Think of gametes. So this is the fusion of the nucleus, which is fertilization per se. And then we'll form the zygote, and the zygote is going to immediately undergo meiosis. Fungi also reproduce asexually and in two ways. So yeasts, for example, which are the only unicellular type of fungi, they can reproduce via mitosis, which would be similar to binary fission in bacteria. So you have one cell that splits into two. We can call mitosis now because this cell has a nucleus, is eukaryotic. But there's another process called budding. And budding is different from mitosis because what happened with mitosis is the cell is going to grow a little bit, is going to split into two, and then grow. These two daughter cells are going to then grow back into the original size. Whereas with budding, you have the original cell here, and then you have a little outgrowth of that cell, starts small and starts getting bigger and bigger and bigger until those two cells split. There is another way for multicellular fungi to reproduce asexually. They can produce asexual spores called conidia. And these spores, remember, asexual spores, meaning that they are produced by mitosis. They are clones of the mycelium that produced them. These conidia, if you look at these pictures here on the bottom, are these little round structures at the tips of this hypha. And this hypha that is upright, so usually the hyphae, the mycelium, is kind of horizontal and really attached to its substrate, right, to its food. But then it would put out this aerial hyphae. These hyphae, they're more kind of vertical. They're kind of pointing up. And then at the tip of these hyphae, there'll be the production of the conidia. So these hyphae, they're upright, are called conidiophores. Phore means to bear. So these are hyphae that bear conidia. Here is an example that you might have seen in your house, in your kitchen. An orange that goes spoiled. Sometimes you have this green, powdery type of fungus. But really, the green here is the spores. And this is penicillium. It's usually green, so you can recognize it. It's the same that we make blue cheese out of. It's the same that we make penicillin out of. And if you would look closely in the microscope, you'd see these structures, which are similar to this one on the left. Now, there's a group of fungi that we call in general deuteromycetes that we haven't seen sexual reproduction. And this is relevant because you're going to see we separate fungi mostly based on the type of sexual spores they produce and the structures that produce those spores. So these deuteromycetes are really hard to classify into a phylum of fungus because we haven't seen sexual reproduction in them yet.